Welcome to the current. This is the North Central Region Water Network's what we call our speed networking webinar series. I'm Rebecca Power, and I will be moderating from the University of Wisconsin today. Since some of you may be new to the North Central Region Water Network, we are an extension-led collaboration among land-grant universities in 12 upper Midwestern states. And we are grateful to have Donna Brandt from the University of Missouri and Francisco Arriaga joining us as our presenters today. And we are also grateful to you all for joining us today and for your patience as we got that link straightened out. So today's topic is soil health parameters and water quality. You'll have a couple of guidelines there on this first slide. So the way that we work these sessions is we want you to submit your questions for the presenters via the chat box, which we've all been using, so you've got practice there. And then we'll have a dedicated Q&A session following our two presentations. And we also, I believe, gave you the link to the phone audio option should you need that. We will also be recording this session, and it will be available both at northcentralwater.org and in eExtension at learn.eextension.org. All right. Oops. So with no further ado, as I said, our two presenters are Donna Brandt, Research Specialist and Lead at the Soil Health Assessment Center at the University of Missouri. She's going to be talking to us about considerations in soil health analysis. So how do we measure soil health, and what are the considerations in measuring soil health? And since we are the North Central Region Water Network, Francisco Arriaga is going to be talking to us about the linkages between soil health and water quality. Are there any? What are they? Stay tuned. All right, so here's a bio for Donna Brandt. I'll let you skim that for a few seconds, and we'll go ahead and get started with her presentation. So thank you, Donna, and go ahead and take it away.
Thank you. Well, as she said, I'm Donna Brandt. I'm the research specialist here at the Soil Health Assessment Center at the University of Missouri, and I've been here about 6 years. That was the time when they were just starting to do soil health measurements, in addition to being a soil characterization laboratory that had been going on for 20 to 25 years. So we had to pick out the analyses we were doing and get the equipment and all of the other information we needed to do the analyses, and we've been doing them for about 5 years now.
Donna, we seem to have lost your audio.
Okay.
There we go. We gotcha.
Okay. So I am Donna Brandt, and I am the research specialist at the Soil Health Assessment Center at the University of Missouri. I've been here about 6 years. We've been doing soil health analyses in addition to characterization analyses that had been done here for 20 to 25 years. So today what I would like to do is provide a perspective on soil health testing, present some considerations in selecting analyses, and provide resources to help you in decision making. So what is soil health? Soil health, it's also referred to as soil quality, is defined as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. So to me, the most important part of that is the function. So what do we mean about soil function? Let's go through some of the different functions that soil provides. It produces biomass, which is what I think most people think of when they think of soil. It regulates water flow and storage. It filters, buffers, and transforms matter. It stores and houses minerals, organic matter, and many organisms. It supports plants and other structures, provides raw materials, and provides clue to past climates, vegetation, ecology, and civilizations. So what I like to tell people is it's a recorder of history. Soil doesn't change its properties very quickly, so what we do now is being recorded for the future to see what we have done with the soil. Soil health testing involves identifying indicators of soil functions that can be measured. So we're looking at physical, chemical, and biological properties of soil. And soil fertility has been looking at chemical aspects for a long time, and there have been physical aspects that have been looked at for a long time. And the biological, the technology is just changing every day. So to me, soil health is looking at how all these interact and looking at them as a whole. So the Soil Health Institute is a fairly new organization that has been looking at some of the soil health indicators and trying to figure out which ones to endorse. What they're looking at is ones that are fairly reliable, fairly consistent, and if they're tests that should be run in a laboratory, is it feasible for a laboratory to run these tests? So just a quick go through the list here: organic carbon, pH, water stable aggregation, crop yield, texture, penetration resistance, cation exchange capacity, electrical conductivity, nitrogen, phosphorus, potassium, carbon mineralization, nitrogen mineralization, erosion rating, base saturation, bulk density, available water holding capacity, infiltration rate, micronutrients are the ones they picked for Tier 1. They are still evaluating other indicators that need further testing to put on a Tier 2 and a Tier 3. Okay, so I'd like to provide a little bit of perspective. An early criticism of soil health was that it wasn't quantitative. So I think we are becoming more and more quantitative, but we have to bear in mind that soil fertility testing has been, according to this article, since 1845. So that's 173 years, whereas soil health measurements have been 10, 20, 30 years, somewhere in that range. So we can't expect to have as much background in all of these measurements as we do in soil fertility because of the variability of the soils. There's 12 different soil orders. They developed in different plant material or parent material, different climates, different vegetation, different sorts of relief through varying amounts of time. Soil texture is different. So what may be healthy for one soil may not be healthy for another. It's sort of like if you were a sumo wrestler, if you weighed the same as a gymnast, that probably would not be good. Or if you were a gymnast, you wouldn't want to weigh the same as a sumo wrestler. So different soils have different, it's more of an individualized measurement. So because of that, I think comparisons are really important, at least at this stage. So if you're going to do soil health measurements, I think it's best if you can set up some sort of comparison. So you may want to compare to a long-term grassland that's on a similar soil that you want to test for soil health, or look at a fence row, and then you could look at those as more of a what's the best case scenario for this soil. Or you may want to compare different managements or different cropping systems, figure out what it is that you really want to know and set up a comparison for that. There are some advances have been made in this area so that there will be a better comparison. There's the Soil Health Management Assessment Framework, which is SMAF. It was developed by ARS. So they have quite a few soil indicators that have been measured on a wide variety of soils. So they have this framework. It's not up on their website now, but you can put in information, but you need to have a lot of the information, soil texture, I think there's latitude, longitude, a lot of different information that needs to go in there so they get an idea of what soils you're trying to compare to. The Cornell Soil Health Lab, they have their comprehensive assessment of soil health. And up until just a couple years ago, they would say that their analysis were mostly valid for the Northeast, and now they have expanded into the Midwest, and you can go to their website and see what they have on there. Our soil health laboratory, we have started gathering this type of information our Department of Natural Resources is providing soil health cost share for cover crops. So they were requiring them to send participants to send soil samples to us. So we've been analyzing these and putting it together so we have state and county averages, and we can expand with that information to be a better comparison. And I think if other analyses, other labs, you can ask them about this, but I think you can still, if you have the comparisons, you can have a better idea of what is good for your soil, or you have a better idea. It's scaled into what you're trying to measure. So this is actually off of this website where it was discussing the SMAF. So I just want you to look at the graphs in the middle, and you will see similar graphs to this on the Cornell website. There are some indicators that are sort of like Goldilocks. You don't want too little or too much. You want it to be just right. So a lot of these types are with nutrients. You don't want to be nutrient deficient, but you don't want to have a toxicity or to have your soil nutrients out of balance. So then the next kind is the more the better. And there seem to be quite a few of the soil health measurements that are like this. Total organic carbon, active carbon, aggregate stability. The more you have, generally speaking, the better health your soil is in. Now there are some where less is more, like bulk density. You want lower bulk density, so you have more pores, so you have a better soil air, soil water, better way to hold and intake water. Another would be available aluminum. It can be toxic, so the less you have of it, the better health your soil is in. So another question is, how good is good enough? Some of these scores are based on a standard curve of the accumulated samples, so they're sort of grading on the curve. So how do we know if say they all failed the test, but you might be the 99th percentile and still not be functioning well enough, whatever that means. We don't know how well do the indicator scores match up to how well this measurement is predicting soil function. So one thing we could look at is crop yields. That could give us an indication on the producing biomass soil function. But I think some of the other soil functions may be more difficult to gauge. You know, if your infiltration rate, your soil porosity, water holding capacity, is it sufficient to provide enough water through the next drought? And that's not a super good question, because if you have a bad enough, long enough drought, you may not be able to hold enough water in your soil, even if it's the healthiest soil in the world. But I think the next question, is it sufficient infiltration to replenish the groundwater supply? That's over a longer term. So, if you have a healthy soil, there should be enough infiltration. The water should get down there. These are just types of questions that I think we need to be asking. Another one would be, are soil microbial populations active enough? Are they high enough to break down herbicides to prevent carryover and to keep the herbicides out of groundwater? These are just some examples. You can probably come up with more and maybe better questions. These are the types of things I think we need to be thinking about and trying to answer. So in just beginning decision making, when you're trying to figure out what soil health tests that you want to use, I think it's pretty much like when you're making any sort of decision or when you're buying something. What is your budget? What are you most interested in? Who is your audience? What type of learners are they? Do they want just straight numbers? Do they want to see more of a hands-on thing, visual, auditory learners? What's the best use of your money? Which laboratories have the most experience with soils similar to the soils that you want tested? You can do analysis in the field or in the laboratory. Some can only be done in the field, and I think you can gather a lot of qualitative information going out into the field. You get people really looking at their soil. Even if they go out to take the samples to send into the laboratory, we get a lot of comments when people bring the samples and things they've realized. I only have 3 inches of topsoil left, or there's this highly variable from one place to another. I think there's a lot of value in getting them out in the field. Some tests are best conducted in the laboratory with the chemical analyses and biological. It's hard to do that in the field. Some can be done both ways, and you need to be thinking about what would be best for your particular situation. If you have a field day, you may want a colorful test so people can see. Again, think who your audience is. Or there may be some of those tests that if you just want the numbers, a laboratory test may be less expensive and may be more appropriate. So there's in-field soil health assessments. You can get the kit. There's instructions online. You can get little sort of questionnaires that just have people go down, like this one here, have them look at the surface cover, what the structure's like. And it's sort of qualitative analysis, but it really gets people started thinking, looking at the various aspects of their soil. There's other tests that, depending on who is using it, it may be more expensive than some people would want, or may provide information that is beyond what the person would use. One example is the PLFA test. It's phospholipid fatty acid analysis. It gives you microbial biomass in addition to what types of and how many of the different types of the organisms in the soil. But if you're doing research, we charge about $50 for this analysis, but you get about 150 data points. So that's 3 data points per dollar. So if you're doing it for research, you get a lot of bang for your buck. Different regions have different needs. Recently, I was showing someone the report that we supply for our cover crop people, and there was a question about electrical conductivity and sodium absorption ratio. And while our laboratory does those analyses, we didn't do them for our cover crop people in our state because these are not issues in our state, but different issues will arise in different regions within the country. There are micronutrients, there may be others as well. Also, different soils may need different laboratory methods. For our cover crops, we used a Bray 1 pH because most of the soils in Missouri are acidic. But if you have a calcareous soil, you would need to use a different analysis. And we do these analyses. You just need to be sure that when you're in that the appropriate analysis is being run for your soil. Another thing to bear in mind is that different labs use different drying and combustion temperatures for soil organic matter. There isn't a standard across the board measurements or temperatures for these things. So if you're going to be comparing, you need to be consistent and use the same laboratory. Another question that people have presented us with, if someone from the desert Southwest was asking about, well, what if there are carbonates and you do soil respiration? So here's an article about that. So yes, if you have carbonates, some of the CO2 given off is not due to cellular respiration, but because there is carbonates in your soil. So you just need to be aware of these things when you are testing. And this kind of goes in with the different regions, but you may have different places or different clients that have special needs. Maybe they have specialty crops, they're looking at micronutrients. Maybe they are not sure what's going on. They may want to do PLFA just to see if it's a microbial thing. Maybe you have clay pan soils and bulk density may be an issue, maybe nutrient ratios. There may be special interests, and you may have to look around to find what analyses would be most appropriate. So for some more information about the soil health assessments, the NRCS website has a lot of information about soil health, and they have fact sheets about all of the indicators, just a wealth of information. If you want to see more about the analyses selected by the Soil Health Institute, this link will take you to their website and they'll give you more information about the analyses and why they've selected it. I'm working with the Soil Health Nexus has a project where we're going to provide an online presence where it will have information about soil health analyses. It will have a toolbox, some information about on-site, on-farm demonstrations and how to go about those, and much more information. So if you'd be thinking of questions, we can discuss them at the end of the talk. Thank you.
Great, thank you so much, Donna. And yeah, so both Donna and Francisco are a part of the North Central Region Soil Health Nexus, what we call our Impact 2020 initiative. So we're excited about that team across the North Central Region and what they're doing. And Francisco's presentation is a part of that. And I also want to echo Donna's shout out, of course, to our partners at NRCS and the Soil Health Institute. And Leslie has put the link to the Soil Health Nexus website, where their regular blog posts and other great information, so you can see that in the chat box. Now, on to Francisco Arriaga here at the University of Wisconsin-Madison and the University of Wisconsin Extension, and he's going to tell us what he's been finding about linkages to soil health and water quality. So thank you, Francisco.
Thank you, Rebecca, and good afternoon, everybody. Thanks for hanging in there with us after our technical difficulties. So what I am going to try to attempt to do here in the next few minutes is to kind of delve a little bit into the linkage of soil health to water quality. There's a lot of interest in soil health for various reasons. Donna talked about those. So here in specific, we're just going to be focusing on water quality. Repeat from Donna, but just to remind you again, you know, the intersection of physical, chemical, and biological properties, that's typically what we call soil health. The way I see soil health, it's just basically all about function, right? Something that we're trying to get out of the soil. With having said that, most of the time we're concerned maybe about growing crops. In this case, you know, we're going to try to delve a little bit more into the water quality side. That's the function kind of we're looking at. So when we're looking at water quality concerns, you know, we can split them in surface water issues and groundwater issues, right? So surface water, things like sediment, phosphorus loading, nitrogen loading, especially from tile ground into surface water. So those are things that we typically are concerned with water quality on surface water bodies. And then groundwater typically is more things like nitrogen, right? Nitrate leaching, that can be a big issue in a lot of parts of the US and the world. Pesticide leaching, and in a lot of places too, depending, especially if you have fractured bedrock, pathogens, either from manure sources or even from septic systems, domestic septic systems, so from human. So the other animal, I guess, is still a manure, if you will. So if those are kind of our concerns, and these are the ones that I'm going to kind of propose for our discussion here today, just to keep it simple and brief, what soil health indicator, what are the potential soil health indicators that might correlate to some of this? And you see that there on the right side. So for surface water, one that is obvious would be runoff or infiltration, right? They're kind of inverse of each other. So you can measure either runoff or infiltration to try to figure out how much water is going into the soil. And that we know it's tightly related to sediment losses and then loading of chemicals in general. So what other things relate? And this is one of the issues that we, I guess, face, which can be a good thing and a bad thing, is that a lot of the soil health indicators and the parameters that we look at, they're connected, right? So aggregation, right? We know that better aggregation should lead to better infiltration. Another one too would be the capacity of the soil, right? That function of recycling nutrients. So if the soil is more effective at recycling phosphorus or nitrogen, we would expect that we would have lower losses. Groundwater, similarly, you know, we have drainage, water retention. So those are things that we can measure, right? And then being proposed as soil health indicators. Again, nutrient cycling. So if we're able to trap the nitrogen in the soil a little bit better and kind of keep it there, relevant, I guess, in the root zone, that decreases our risk for nitrate loading into the groundwater. And then pesticide breakdown, like Donna mentioned earlier too. So and those are things that have been studied individually. So that's one of the problems we're seeing too. And you'll see here in a second here as we can move forward is there's a lot of data out there. The problem is that a lot of times, obviously, it's not tied into soil health or soil quality, but or it might not even be tied into water quality. So there's a wealth of information, but it's I guess the challenge right now is try to tie things together and identify what knowledge gaps we're missing. So the general idea would be this, right? So we have a soil health in the bottom axis here on the x-axis, and then water quality on the horizontal, vertical axis. And I have there 3 proposed responses, if you will. So the idea here is that as we increase soil health, we should improve water quality. Is it a linear plateau? Is it a linear relationship, linear plateau, or a quadratic type function? We really don't know. So that's something that I guess we need to think about. And here I'm not talking about individual soil health parameters, but kind of as an aggregate. I don't want to use the word index, but it's kind of a soil health as an aggregate, not individual property. And here's an example of this interconnectedness of things. So if we're looking at here on the left, organic inputs added to the soil, right? We know that they're going to break down. Some sugars are going to be forming, right? Some polysaccharides, and that can lead to aggregation and infiltration. And we know that these two are interconnected. There's other components of the organic material that will affect these, right? So if we're looking at indicators, what indicators we want to measure for, let's say, thinking about runoff, we could potentially measure aggregation or measure infiltration. So the question becomes, what is easier to measure? Donna was talking about this issue, right? What is most relevant for your condition? So it becomes kind of a at this point yet, I think it's a kind of a personal preference in some sense. And the other thing too is that there are feedback mechanisms. So on the example before here, it's a biology that's making this happen, right? It's affecting the physical property of the soil, but the physical property can also affect the function of the biology of the soil. So in this case, we have different bulk densities, which are this different lines in the different colors, so increasing bulk density. And here we have basically carbon mineralization in the soil. And as the bulk density increases, that means the porosity of the soil decreases. We see that all of a sudden we have a reduced potential for carbon assimilation into the soil. And that's related to the porosity of the soil. Like I mentioned, that's the habitat where the biology of the soil exists. So that kind of, I guess, highlights, the issue between these parameters that are so connected. How about enzyme activity as an indicator? So Garcia and others did a pretty neat report where they look at dehydrogenase activity as an indicator of soil degradation and microbial activity. So they looked at 18 different soils, and one interesting thing they found was that soil organic matter content, so the total soil organic matter, was not related, but the dehydrogenase activity was positively correlated with soil respiration and biomass carbon. So what they postulate here is that in degraded soils, the organic matter, it's more stable organic matter, so the biology cannot get to it as readily, and that's why they were able to see it as a potential indicator, right? So this is the ability of the soil to assimilate materials. All right, so talking about water stable aggregates, and so here's an example looking at no-till and conventional tillage long term, and we're looking at basically here the impact of fungi on this. So now looking at the biology, can we use maybe some kind of fungi assessment or water stable aggregates? What is their connection, right? So on the left column here, we have aggregate size, so going from bigger aggregates down to small. And then what these researchers did was that they had a control for their soil measurements, and then they had the same tillage, and they apply fungicide to eliminate the fungi and kind of isolating the activity or the impact, if you will, of the fungi on water stable aggregates. And so the first thing to note is these asterisks denote statistical significant difference between the tillage overall, so the tillage main factor. So there was a significant effect between tillages and aggregates greater than 2,000 micrometers, then between 250 and 106 and less than 53. If you look at the no-till, there was a significant difference. So there was a significant contribution from the fungi in generating water stable aggregates, where in the conventional, it didn't matter. The fungi were disturbed enough that soil was disturbed and disturbed the activity of the of the fungi, and they were not able to affect water stable aggregates. So another way they were able to show that was actually looking at total carbohydrate and then the mannose-xylose ratio. And here you see an example. So this is another potential indicator, right? So you can look at total carbs. We know that it's relating to the water-stable aggregates, right? And so we're trying to make that connection. So this is another potential indicator, right? Interestingly here that they didn't find a difference in soil respiration between the tillage measurements. All right, so we're talking aggregation. So well, how is that relating to the water quality? And potentially in this case, we're going more after runoff. So here's data from Franz Luber looking at long-term no tillage and long-term conventional. This is similar soils to the prior study that I showed you a second ago here with the fungi. And so what Franz Luber did was that he looked at intact soil and then soil that it was sieved. And so the purpose of that was that he looked at long-term disturbance of the soil and then short-term disturbance of the soil. So I'm showing you here a kind of an average. He measured these over time. So here in the top here we have infiltration. Again, no tillage on the left, and excuse me about that. No tillage on the left and conventional on the right, right? And we see, again, there is a big difference in infiltration. This is in centimeters per hour between the conventional and the no-till. But if you look at the disturbance, right, the long-term disturbance, there's a significant difference between that. But then if you look at within tillage, there is a big difference in the no-till. So you have good aggregation to disturb it, and you're losing that capacity to infiltrate the soil, where in the conventional, it did not matter. He also looked at the portion of water leach, so that you could see this as drainage, so potentially what could get to groundwater. And there was a big difference, right, between the no-till intact soil and the conventional. So a lot more water leaving the conventional. So here we see that soil under that management would have a greater risk of groundwater contamination, if you will, nitrate or pesticides. And so he was able to link that then to water stable aggregates. So we're going back to the aggregate. So the aggregation, if you look in this figure, this is with depth, soil depth, and then water stable aggregates on the horizontal axis. The circle is the conventional till, the square is the no-till. And so you can see there's a lot more of those squares here. So that means there's more water stable aggregates with the no tillage scenario. So here we see a direct relationship between aggregation and infiltration, and also the leaching. So we can kind of see then what indicator would you want to measure, right? You measure infiltration directly. The other thing too, who's your audience, right? So you're going to farmers, you're talking about aggregation, they understand that, but if you're talking about runoff with them, maybe infiltration is more appropriate indicator to look at when we're talking about water quality. Here's data specific to a research project that a former student of mine conducted here in Wisconsin, where we're looking at cover crop. We're using cereal rye as a cover crop. And we did rainfall simulations 3 times within a year. And there's the reason I want to show this is that there is a kind of a seasonality to some of the measurements that we do. And this is just true also if you collect a sample in the spring versus fall. For a lot of these indicators, soil health indicators, there's going to be differences. So that's one challenge that we have. But overall, we see that there was a positive impact in reducing runoff with the cover crop. We also saw that there was also overall a lower phosphorus loading, so lower losses of phosphorus in the surface runoff. And so if you look at here now, the average of those 3 simulations, and we're comparing now to water stable aggregates at the bottom, and this is the no cover and cover crop. So we see that the cover crop did reduce the runoff and also the total loading, phosphorus loading, and that was tied into the aggregation, right? So that's again showing that linkage and the potential for those measurements as indicators. This is after 3 years of the cover crop. And if you want to do things more in detail, like we do in research, you can see here how the aggregate size distribution. One thing that was interesting was that for the phosphorus loading and for the runoff, tillage did not have a significant effect. But when you look at aggregations here, or aggregate size distribution, these are dry aggregates, that actually you can pick up some differences So that's another, I guess, interesting thing and kind of maybe warning in a way, right? That sometimes we might pick up a difference in indicator, but it might not necessarily translate into a function, which in this case would be the water quality. And again, this is looking at water release, and it's kind of a similar point I made before. So this is water retention, looking at tillage, and we were able to pick that up with the water release, those differences, but again, not really translated to the runoff losses. So the challenge I think we see here is that we can, like Donna pointed out, that we can do contrasting management systems and changes over time. I think that's possible right now at this time. But because we have such a range in soils and climate within the country or within even a region, not even to mention difference between the world, developing thresholds for indicators, it's actually the main challenge in my view, right? So if you measure aggregation and you have a certain amount of, let's say, 60% water stable aggregates, can we definitely say that once you get that to that point, we have significantly reduced the potential for phosphorus loading, right, to surface water? Or if we can measure the water release curve and we get to a certain criteria, can we say with certainty that we are reducing the potential for groundwater contamination? There are a lot of other challenges I think that we're going to have to bridge before we get to those thresholds. For example, trying to figure out what quote-unquote universal indicators or what properties to measure. I think there's still a lot of debate about that in the scientific community. What is actually the most sensitive? And it might be that a soil in the Midwest is different to a soil in the Southeast or out west or in other parts of the U.S. And also establishing specific measurement procedures, like Donna talked about, so that matters. And also another thing that is very important, the sampling protocol. So that timeliness, I showed you that difference in the effect of the cover crop with time of the year, right? So we see the same thing with the soil health indicators and soil properties. So setting up some protocols and be able to adapt them in several regions. And then eventually is development of the ROS linkages between soil health factors and water quality. I think there's still a lot of work that needs to be done. There's a lot of data out there, but I'm sure we're going to identify gaps in that knowledge base, and that's another potential area of research for researchers. So with that, I think that's all I have. Rebecca?
Great, Francisco. Thank you so much. And we have some questions coming in here. We're going to get right to those. I will ask Francisco and Donna, are you able to stay for 10 minutes or so if we extend the time of the webinar for 10 minutes?
I am.
Yes.
Excellent. Great, thank you. So we'll just do that because we started a few minutes late. And we'll go right to Aaron's question. And I'm going to read this just for the value of the folks that will be accessing this presentation, a recorded presentation. So Aaron from Wisconsin DNR asked, do you have any thoughts on trying to come up with something like a soil health physiochemistry-based ortho-P, dissolved P retention metric? Or do you know something like this in the works in the soil health world beyond something like traditional soil test P? He's saying he agrees that while infiltration aggregation seem like a logical metric for total P associated with soil health particles, accumulation of organic matter also concentrates a potential for microbial oxidation and mineralization of that organic matter and mineralization of P leading to dissolved P. Let's see. So this might have implications for water quality, even if it does infiltrate or move to shallow groundwater. So thoughts about that from our presenters?
So Aaron, thank you for your question. Absolutely, I agree with you. So one I guess thing that maybe I didn't highlight or mention, I guess or emphasize was how interconnected a lot of these parameters are. I'm not aware of any similar measurement in the soil health world related to, like, say, water trackable phosphorus and trying to link that to phosphorus losses. But that's a good idea. I think what we're going to find out in the long term is that it's not going to be just one measurement that we're going to make for a certain function that we're after, we're going to have to probably measure several things, and it's going to be kind of a combination because a lot of these things are interconnected, and one indicator might not completely capture the function that we're after. So I agree with you. There's a lot of things that we need to sort out. So these are very good points.
Donna, any additional comments?
I don't think so.
Okay, all right, great. And Les has another good question. Les Everett from the University of Minnesota. You reviewed several experiments. I think this is for Francisco. However, duration of treatment in years weren't usually mentioned. Isn't the duration of treatment a significant factor in effectiveness?
Hi, Les. Yeah, absolutely. The management, and I purposely tried to stay away from management factors, and I was trying to just focus on the soil health indicators. Yeah, the length of the treatments makes a big difference. The data I show you from the rainfall simulation, the last study that I show you, that was after just 3 years of the tillage and the cover cropping, and that's on a corn silage system. The other ones that I show you, talking about the fungi and the aggregation that work that Franz Luber did, those were long-term management systems as far as the tillage, greater than 10 years. So yeah, the time of the management makes a big difference. And so that's why I think Donna was making the point of measuring or comparing management systems or measuring things over time. That's something that we can do, I think, right now to look at soil health parameters.
I would just add that it takes a long time for it to change, probably about 5 years before you see a lot of difference, also depending on how much change has been made, but definitely I agree with Francisco.
Excellent, thank you. And again, sorry folks, I know some of you are going to need to go at the top of the hour, so we'll obviously have these answers recorded and available to you should you want to check back at northcentralwater.org. We'll go on to Eric Hurley's question. Some researchers in Ohio assert that increased infiltration and preferential flow pathways due to no-till has increased dissolved reactive P and drainage water reaching Lake Erie. What are your thoughts on this?
So Eric, so I'm assuming under that scenario, we're talking about tile drain. And yes, in those scenarios, you can have that. And this is one of, when I go talk to farmers, one of my favorite things to say or says is that there's no free lunch. It's all shades of gray. So it depends really on the scenario. It's very site specific. So if you have tile drain and you have preferential flow to those tiles, yes, that actually will represent or can create a direct pathway into surface water.
Okay, and forgive me if I butchered this name, Sangir Kabir. Can you send out the reference cited in the presentation, especially the experiment using fungicides? So Francisco, if you can post that while we're working here, get that in the chat box, that would be great.
Will do.
Awesome. And then Adam, to date, has there been any research that shows zero-till and or cover crops having a negative impact on soil health and water quality?
Sorry, I'm trying to multitask and I'm not good at multitasking. I hyper-focus, not multitask. Research shows zero-till or cover crop having negative impacts on soil health and water quality. Not on soil health, Adam. Water quality, there's been some work a little bit related to the phosphorus and the stratification. And so I think that's going to be very depending on the soil type and also how old is that system, that no-till. And again, there's no free lunch, there's shades of gray. So we've been conducting some research looking at winter manure applications and looking at tillage. So no-till in a fall chisel plow system, and we're measuring runoff in the winter. And so one of the things that we found in this scenario, these are really nice prairie soils, highly irrigated on a natural way, so they have awesome soil health. We are actually seeing more runoff out of the no-till than the chisels, the conventional tillage. So it all depends on the scenario. So that there can be some situations that the management, I wouldn't say necessarily the health of the soil, but the management might not react the way that we're expecting it.
I was just going to mention that I think in some cases there has been at least a temporary increase in bulk density that I might expect. You know, it just depends how long they've been doing the different, doing no-till, or what kind of comparison you're making, what time of the year you're measuring.
Great, thanks for that addition, Donna. And glad to know that Les is still square dancing.
Good for you, Les.
Good to check in on the really important things in addition to soil health. And Eric's next question, Eric Hurley, will improved soil health reduce nitrate, reaching tile water or groundwater? What's the mechanism, and does it always work?
Will soil health reduce nitrate leaching on the tile? To tile or groundwater, right? So I'm looking at two different ways. So for tile, let me get to a topic that sometimes gets controversial. So I think a lot of times in general when we're talking about soil health, we get a little confused with the end in mind, right? We're trying to improve the health of the soil, and we get confused with the specific management. I think, and not everybody thinks this way, that you could probably reach to a soil, depending on your conditions, where you are and your scenarios, you can reach to a soil that has better soil health that you currently have without necessarily maybe using, let's say, no tillage or even cover crops, right? Those are management. Those are the tools that we get where we want to go. So it's going to depend a little bit, right? So soil health, I think in some scenarios can reduce nitrate leaching. So if you're using a cover crop as a trap crop, that's going to improve your soil health, but it's also going to have then that benefit of recycling that nitrogen. Same thing with the tile, but then like I mentioned earlier, if you have a highly structured soils and you have direct continuity from the soil surface, pores, macro pores straight to your tile, you very likely will see nitrogen being lost through that tile to a greater degree, or maybe even to groundwater, depending how shallow that groundwater is. On the flip side, let's say a pesticide, maybe you have better biological function, and that pesticide can be degraded. So again, it's I guess pretty complicated, right?
Well, then for both of our presenters, that gets me to a question that's been on my mind. So given the complexity of the relationship between soil health and water quality, you know, when you're working with farmers or farm advisors of various stripes, whether it's, you know, NRCS, district conservationists or extension folks or crop advisors, you know, whoever, what do you tell folks about the relationship? You know, like if you had to start that conversation, how would you start it?
Renee, you want to take a crack at that first?
Did you say you were going to take it, or you want me to take a crack at that first?
Yeah, do you want to take a crack at that first?
So the question is just how related are soil health and water quality?
No, it's so if you're talking to a farmer or a farm advisor, no matter who they work for, folks that are working with farmers and are talking about this, the value of soil health, specifically related to water quality, what do you tell them, or how do you start that conversation, given the complexities that we've got here?
Well, I think I would just start with, talking about what happens as you can increase soil health with the aggregation. It's the soil particles are sticking together, so they're not going to come apart when the rain falls or when it stays wet. So you're not getting the erosion and the potential of those particles leaving the field to add to surface water. Talk about infiltration, the water going in and being able to be filtered and being acted upon by the microbes. I think I would just go through all the parts of it and how it all interacts. So Maybe Francisco has a better answer.
No, I think that's you're spot on, right? We need to look at it as an aggregate, not individual things, right? Soil health overall, it's a good thing. We should strive for improving the health of our soils. You know, they makes them more resilient, right? Somebody has here comment that Adam, that a healthy soil is more resilient to water quality challenges. That's true, and also to water quantity issues, right? The drought resistance and all that. So it's looking at that aggregate. So I think soil health, you know, when I go talk to farmers, I think it's a great conversation starter, because you can start talking about individual things, but then you then bring to the picture to the whole system. A lot of times we just get so focused on just one individual item that we forget kind of the whole picture. We forget about the forest, and we're just looking at the trees. So I think it's a great conversation starter.
Excellent, excellent. So, and we know, you know, this presentation obviously has given a lot of us that are doing research in this area some great ideas and great things to work with. And there's always that communication challenge when something this complex. So it's helpful to hear your perspectives on that as well. Let us know if you have any further questions, folks. On the line, and compliments, of course, are always welcome. So thanks, Jill, for that hurrah there. So we just are completing our Q&A session, and here are the email addresses for our presenters. So we love to introduce you to folks that I know a number of you are already working with Donna and Francisco, and some of you may not be familiar with their work. So we're happy to introduce them to you in that case. And we'll go ahead and let you know we've got a couple of, so to access this session online, you can go to northcentralwater.org, and we have a webinar tab that you can find our sessions. We also have a couple sessions coming up that we want to let you know about. We have another one of our teams, the North Central Climate Collaborative, their June webinar, June 25th at 1 p.m. Central, understanding the influence of climate information on farm decision making. So there are definitely some soil health tie-ins there, as you can imagine, and you can register at northcentralclimate.org. And then our next current webinar for this series is on July 11th, and the focus there is on aquatic invasive species and successful efforts to manage aquatic invasives. So we'll have Doug Jensen at the University of Minnesota Sea Grant, Tim Campbell from the University of Wisconsin Extension, Greg Hitzroth from Illinois Indiana Sea Grant, and Paige Felice, who's a research assistant with the Department of Fisheries and Wildlife at Michigan State University and Michigan State University Extension. So thank you so much again for hanging with us with that technical difficulty in the end. Thank you so much to our presenters. These folks are really on the leading edge there of what we're doing with soil health and helping people be able to effectively use that concept for management, and also, of course, linkages to water resource management, water quantity management, as well as water quality. And finally, thanks again to all of our participants, and we look forward to having you with us again in future months. Take care and have a good rest of your day.